Thanks, Atlanta, for the kind introduction. So today I will be talking about the CCD2, Crystallization Constant Designer 2. And well, this is a tool that allows you to quickly design expression constructs, either for crystallization or for other techniques. So in short, whenever you are in a structural biology project, you have proteins that you want to analyze the function of. And what you will end up doing is most of the time design some truncations of those proteins, then clone them, express them, and do your assays downstream. Now, the process is repeated typically tens to hundreds of times, and it's not particularly difficult, but it's quite boring and it's quite time consuming. And in particular, to design truncation, you need to run your sequence to multiple analysis and then somehow take the disparate outputs of these servers and put them together in a single view so that you can decide where to cut. And then to clone, you need primers. And to design primers, you need to go from the protein sequence. Let's say you want amino acid number 30 as a starting point. Go back and translate to a DNA sequence, which means it will probably be codon 180, or is it at 181? Who knows? And then you have templates, primers, overhangs, and so on and so forth. All of this is simple, but boring. And what CCD does is it allows you to find the DNA template for your PCR, if your protein is in UniProt. It gives you an automated protein analysis, and it also allows you to choose your constructs on the protein sequence. You don't need to think about DNA anymore, because at that point, CCD will design the primers and give you useful information about your constructs without you ever needing to think about it. I have to say that it's called CCD2 because it's the version 2 of the CCD that was designed a long time ago. But this tool was obsoleted by browser technology. And so me, for the back end of the server, and George Damascus for the front end that works in your browser, have decided to rewrite it and also add quite a few new features. I will now go through the first part, which is how we show you the information that we think you need for protein construct design. And so for that, I will try and say what really we mean by construct design. So at a glance, in a very, very simple-minded way, proteins are made of domains that are connected by disordered regions. So whenever we cut within domains, we get typically unfolded proteins, which is insoluble. And so the first rule for designing construct is do not cut within domains. For the rest, disordered region can be your enemy if you're doing structural biology, because conformational freedom is the bane of all methods. So in that case, you want to remove them. Otherwise, if you're doing functional studies, you might want to keep them because they contain important functional sequences, or they might even be intrinsically disordered proteins. Whatever the case, you can generally distinguish a domain from a disordered region by somehow their signature. So a domain tends to have a secondary structure, a hydrophilic, hydrophobic mix of amino acids. It is usually evolutionarily conserved, and you can typically find signatures in signature databases. Disorder tends to have no structure, fairly low complexity. It is more evolutionary variable, and it also can have some signature sequences. And so what CCD does is it aggregates a series of very quick, and the emphasis here is on quick rather than accurate, so that we can give you the answer in 5 to 10 seconds. It gives you a very quick series of predictions so that you can then decide what's domain and what's disorder. The prediction that it gives you are evolutionary conservation via an ensemble or blast alignment, a quick overview of secondary structure prediction with a consensus of 4 simple methods. It also looks at disorder with amino acid composition, and then it looks at signatures for domains via the SMART database of protein domains, coils, nuclear localization signals. And finally, it also blasts your sequence against the PDB to see if any of sequence or part of your sequence has already been solved, which of course indicates the presence of a domain. And finally, it also looks for phosphorizational modifications via phosphosite although this only works for mouse and human, which are the ones that have experimentally validated modifications. So I will show you later how CCD works in a demo, but because we're doing things online, it's easier to simply paste things into a slide so that I don't give everybody seasickness with scrolling. So this is a zoomed out version of the output the CCD would give you in terms of sequence analysis. This here is your query sequence. On top, you have an alignment, or at least in this screenshot, part of an alignment. And at the bottom, you have the predictions I talked about before. Now, as an example of how to use CCD, I can sort of talk you through how I would work with CCD in order to understand a protein that has little no annotation, such as DEF6 of human. So first of all, I would look at the secondary structure prediction. So the first 4 lines are secondary structure. And as you can see, there is secondary structure predicted all the way from the N-terminus down to here. Then there's a break, there's possibly a helix, and nothing else. So this tells me that most likely there's a somewhat globular N-terminus, break, small helix, and disorder. Then I would move on and look at disorder predictions. Red is disorder, green is predicted globular, and white means that we do not have any prediction. If we look at this, we can see that this particular block of secondary structure seems to be connected via disordered region, so it's possibly a little floating domain. There is also disorder around the helix, and there's possibly some disorder here in the middle of this long predicted helix. Then I would go down and look at the signatures, and you cannot really see it right now, but the smart database tells us that there's a pH domain, so that's a plexstream homology domain. And down here, blue means we get a lot of results. There are a lot of results in the PDB search that tells us that there is a domain here. If we look at the coil-coil prediction, which is this line here, there is a very strong dark blue prediction that goes all the way to here. And if we look at the nuclear localization signal, the predictions are very, very marginal. So overall, I would say ignore the NLS. It seems just an artifact. There is a pH domain with an N-terminal extension because this part is not really identified by the SMART. And then we have a coil-coil at a C-terminus. Then I would go on and look at the conservation. And we take our alignments from ensemble. These are alignments of fairly close sequences. So on average, for conserved parts, you would expect a lot of coloration, and this is cluster X coloration. And for divergent parts, you would expect only, let's say, half to 30% color. So if you look then at our alignment, you can see that there seem to be some homologs that starts from the pH domain and are missing almost entirely the N-terminal part, suggesting that this is indeed a separate domain. And then you also see that there are gaps. These gaps you do not see, for this one you see it, for this one you don't. It means that in the alignment, there is at least one sequence that has a long insertion there. And if a long insertion is possible, it means that this is a flexible region. And so it means that you can potentially also cut within that. So all of this suggests that the coil-coil is split by a long loop. And so it's actually 2 little coil-coils that possibly are parallel or anti-parallel. Finally, if you look at the post-translational modification, there is a strong signal of phosphorylation down here, more or less where the helix is, which suggests that this helix might be some sort of, functional motif. So overall, if I were to cut, I would cut the full length. I would remove the helix and test with and without. And then potentially for crystallization, I would cut away the N-terminal domain to remove the flexibility. Cuts in here and here are also possible. So that's how I would design constructs. And now, fingers crossed, I'll try to go to the live version and show you how the primers are designed. So of course, we need to start from a DNA sequence. You can start by pasting your DNA sequence or simply pasting in a UniProt identifier, and CCD will look into the UniProt entry and determine which isoforms exist and what DNA codes for each isoform. It typically takes only a few seconds. So then you can show what is the alignment of the isoforms. to see if you can spot some biologically important insight. And you can also see which DNA codes for which isoform by opening that. You then select the isoform you want and click submit. So then CCD will run its predictions. Again, the emphasis is on trying to be as quick as possible. And then supposedly you know already where your constructs are, or you designed them as I showed you before. And all you need to do is to mark where you want your construct to start and where you want your construct to stop. And here I'm clicking just randomly. Once you've done that, you have to decide what sort of primers you want. And there's two parts to that. The annealing part, so the part of the primer that anneals to the sequence, you can specify a melting temperature or a base length. Note that this applies only to the annealing part, so your primer will actually have a higher melting temperature. And then it's the overhangs. So most people will tend to use the custom function in which you specify your own custom overhangs, which can also include known DNA characters in case you want to specify phosphorylation for ordering the primers. You can also specify restriction cloning, which allows you to put a start or a stop codon, a restriction site or a custom restriction site, and some extra bases so that your enzyme can actually grab the restriction site. Or if you are running our server, you can use our NKI-linked vectors, which have already pre-programmed in hard-coded overhangs. Now, we are planning to distribute the code, so eventually, if you decide to run your own version, you will be able to program in here your own vectors and get your own overhangs just at the click of a button. Whatever overhangs you want, you can then give your primer a name, hit get primers, and then select and copy-paste your primers in the ordering form of whatever company you use to order primers. You can also save this as a comma-separated value. In addition to that, CCD will also give you the details of all the possible constructs that can be produced by these combinations of primers. So it will give you the protein sequence, and it will also give you the molecular weight and the estimated isoelectric point and the estimated extinction coefficient. These are calculated with proc_param. If you have your own vectors coded in, or if you use the NKI vectors, you can also choose which vector to subclone things virtually in, and then show tagged or cleaved constructs. So this will give you the sequence data for the tagged version of the construct and the cleaved version. This is a GST his, so it gives you both cleavages, either the GST alone or the his. Again, this can also work with your own vectors if you run your own copy. And finally, you can also subclone things in an NKI vector or your own vector. And then this will give you the DNA map so that you can open it to check your sequencing, or you can open it to make plasmid maps in your own DNA program. So in a nutshell, CCD, give it a DNA, decide where your constructs are, and what's important, click on the protein without thinking of the DNA at all. Give your primers a name, choose an overhang, and you can get your primers. It's all very, very quick. One final note, since we've been graced with an extra couple of minutes, the alignments. So the alignments, we try to give you as much as possible alignments from Ensemble. These are pre-calculated alignments that can be retrieved very, very quickly. And most of the time, they are also rather large alignments. So in order to make things fast, we try to give you only a series of representative species for the alignment. This is because otherwise you will explode and it will take a long time. But if you want, you can simply untick this box and then you can get the whole 300 sequence alignment by hitting realign. This might take multiple minutes. Additionally, you can save your alignment as FASTA file, which is very, very useful because, for example, this alignment can be used as an input for HH-Blitz as a SID, or you can use it to save for your own data. So there, we also have a tutorial, up here, and there's more information in that I can give you in this talk. And with this, I am in the acknowledgements. So as I said, I wrote the back end of the program. George Damascus wrote the front end, the part that is in JavaScript for your browser. The project is from Tassos Perakis, and the funding came from here. And you can find the CCD at this URL, or Google CCD plus NKI. Otherwise, you get charge coupled devices. The NKI vectors are also very useful. You can find them at AdGene or via an MT8 at the NKI protein facility. And if you have any questions, feel free to contact me. And that's it. Thanks for the organizer for giving me this opportunity, and I'm ready to take questions.